This is InfoHealth starting up. So this is the September session, and we're doing it electronically today and probably the next two. Anyway, so this one today is about fixing genes, and next month is a very important one. It's about Alzheimer's disease, but especially with the focus on new drugs. And one drug in particular for Alzheimer's disease, which was just approved by the FDA in the United States, but that decision was a very controversial one. So that was the reason why I inserted that into the October slot. And then an unusual one early in November to talk about miracles. And it may be a miracle that I'm talking about it, but it's an interesting subject. And just so that you remember that beginning in early November and running for 6 weeks, this will be the 5th year that we review the Nobel Prizes. And that's turned out to be quite a good series. And I think it gets stronger as each year goes by, because we learn more about the nature of Nobel Prizes, but also there are certain themes that recur with the Nobel Prizes as well. So looking forward to that. So that'll be what, 4 weeks in November and 4 weeks in December. And then the InfoHealth returns in its normal format, meaning that the McMaster University Medical School, the Niagara branch, will be involved beginning in January. So we're back to normal. We'll see what normal is, whether that's live or not, if we feel comfortable with that, or whether that'll be electronic as well. I don't know. We'll see. So just to begin, the first slide is Homo erectus. So that's 1 million years ago in Kenya. And I like to start out all of my sessions with something on this theme about the origins of humans, and something that we'll turn to in the new year will be the relationship between science and art and religion. So that's what's coming up. Now, here are 2 books to look at. One, probably the bestseller is the one on the right, The Codebreaker, written by Walter Isaacson, who did a masterful biography of Albert Einstein, and a very good one on Leonardo da Vinci and Steve Jobs and several other people. I personally don't think this was his strongest one, but it's about Jennifer Doudna and the CRISPR gene editing technique, for which she got a Nobel Prize last year. So that's one book. The other one is one that she wrote in partnership with Samuel Sternberg. I think it's 3 or 4 years before she won the Nobel Prize. So it's pre-Nobel. And it's very good. So the title is A Crack in Creation. I wonder if you've noticed the highlighted letters there, in yellow. Those are the bases. So alanine and cytosine and alanine and cytosine again, and et cetera, through those words. So those are all bases in DNA. So just moving on here. 2 more books of interest. Gene editing in general, but CRISPR specifically, are certainly in the news for their potential for editing humans, and the risks and the ethics that surround it. Henry Greenlee really has done really quite a good job of this. He's a Harvard prof, and that's certainly a good read. A really good read is the one by Philip Ball on how to grow a human, and it'll blow your mind with some of the concepts in that book. So those are two sources. So the next slide just shows what's coming up. The anti-amyloid drugs for Alzheimer's disease, miracles I alluded to in that Nobel series. Now, here's a picture of DNA, and on the far left is the famous double helix. Although I will say that in 1953, with their canonical article in the journal Nature, by the way, that was only about a page long. The diagram was much simpler than this, but certainly highlighting the double helix and hinting at that the complementary bases, these are the central things connected. So DNA has been likened to a ladder, a twisted ladder, if you like, and the sides of the ladder are made up of phosphate and sugar groups. So there's S sugar and phosphate. So those are the sides of the ladder, the twisted ladder. And the sides of the ladder, the rungs, are created by complementary bases. So thymine and adenine together, always. You can reverse the order, but those two always go together. And so do cytosine and guanine. You can see that in the more kind of traditional chemist format on the right here. But there's the double-stranded DNA that figures large in today's talk. So that's a very basic slide. Now, just some points about genetics as of 2021. Well, for some time, the facilities have been there with genomic sequencing and genetic tests to assess genetic risk factors for diseases like multiple sclerosis, cardiovascular disease, and for that matter, the dementias. And sometimes it may be just a single gene responsible for the disease, as it is with Huntington's disease, and that's an autosomal dominant disease. If you carry that gene, you will 100% develop Alzheimer's disease. And then the next two, gene therapy and gene editing, they're not quite the same, but we'll discuss those two a little bit later. In vitro fertilization has been a tool in our hands for at least 20, well, more than 20 years, probably closer to 30 years. And with the advent of being able to sequence the genome, then if the IVF procedure harvests several embryos, each of them can be tested individually to see whether in fact they carry normal copies of whatever gene that you're worried about. So that liquid biopsies, this is an interesting thing because every cancer, well, can't say every, but most cancers shed cells, and the cells end up in the circulation. And those bits and pieces of those cells often carry DNA, and that DNA can be sequenced looking for specific cancers. And it's a technique that can be very specific and very helpful, very sensitive. It's not mainstream yet, but I think it's going to become so. And just skipping the genomically directed cancer treatment for now, genomics has really changed a lot, because now it's possible to trace DNA or track it or restore it anyway when it's several hundred thousand years old. In fact, I think the oldest is 1 million years now for a mammoth, or at least for any mammal. And it's certainly been a way to trace sex between species, especially archaic humans, Neanderthals and Denisovans with humans, or between each other. And it's also a tool for tracing migration patterns and tracing ancestry. And the interesting thing is we don't have fossils for some species that are called ghost species, meaning they show up in the DNA, but we don't have any fossils to match. And we don't know what that ghost species actually look like. Now, just a few words on genes. Genes have addresses. I live on a street. My house has a number. I have an address. Genes have addresses on different chromosomes at different places on the chromosomes. And each gene has a start and a stop identifier at the beginning and the end of the gene. Genes are also, many genes are collaborative. They work in societies, groups of them. And for complex traits such as height, and especially for cognition, hundreds, if not thousands of genes are involved in some way or another. But they work often in cooperation, and those that work together most commonly or the most interdependent on one another tend to be grouped in the same chromosome and in the same region of that chromosome, but not always. A single gene may have different functions depending on the company the gene keeps. Many genes have very ancient heritages and similar functions in widely different species such as the worm, fruit fly, a mouse, a chimpanzee, and humans. The genes may be very, very similar, perhaps not identical, but very similar. Humans with heritages outside of Africa contain about 1 to 4% of Neanderthal genes. And in Australia, Aboriginal peoples carry about 4 to 6% Dystrophy DNA. I won't talk about bottlenecks, the founder effect, the genetic drift right now. Now, some more about genes in general. Humans possess about 25,000 protein encoding genes. Now, that's not all, I mean, the total number of genes is much larger than that, but those are the genes that are responsible for protein. Cells make around 100,000 proteins. That's a really an extraordinary number for single cells. Some proteins are responsible for activating or suppressing genes. And one example of which is normal cellular differentiation that follows the first division in a single fertilized egg to, over the subsequent weeks and months and few years, to form hundreds of different cell types in the body. Now, isn't that extraordinary? They all have the same genome, but obviously not all of those genes are active at the same time. So differentiation and maturation in all species is a carefully choreographed sequence of activation and silencing of various genes and gene combinations. Conservation of genes over long periods strongly suggests that they were and continue to be vital to normal cellular function. A Nobel Prize was awarded for oxygen sensing genes and their protein products a couple of years ago, and that was certainly the case with those essential functions that are shared across many, many species. So no surprise about that. But what about mutations? Now, we're in the middle of COVID pandemic, and we're well aware of different variants, and we've heard about different mutations occurring. Well, every time a cell divides or a virus multiplies, copying errors occur. Now, most of those copying errors are neutral. That is, they don't have any effect. Some of them are lethal to the virus or to the bacteria or to the cell in our body, and that's it. And a few confer some advantage, whether they spread throughout the species or not, or if whether they're adopted so to speak, in subsequent generations. That really depends on a whole lot of other factors, like such as natural selection and chance. But still, some people have likened mutations in genetic material to monks copying the Bible, and the parallel would be there are about 6.4 billion bases in the human genome. It's bound to happen with each replication of the DNA during lifetime or with sex, that some changes will be introduced, some errors introduced, chance errors. And likewise with the Bible. The monks made mistakes, and so did the monk editors make mistakes. Interestingly enough, cells do carry a spell check that picks up many of these mutations. It corrects them, but not all of them. Most errors involve a single base. A base might be deleted, an insertion of a base might occur, or other changes might occur. Sometimes triplet repeats occur, meaning 3 bases in a row recur again and again and again and again and again, many times over. And that happens in Huntington's disease and a myotonic dystrophy. And if you think about it, the coding for proteins involves triplets of bases. So 3 bases in a row code for a certain amino acid. And amino acids are the building blocks of protein. Now, if you have a series of repeats of the same triplet, repeating again and again and again and again, then you end up with a protein that's badly distorted, so that the same amino acid might occur 20 times, 50 times, 100 times. And the severity of the clinical disease in myotonic dystrophy and the age of appearance of Huntington's disease is in part related to the number of triplet repeats. Now, here we come to the central dogma of molecular biology. This is simplified. So how do we get from DNA to a single strand of messenger RNA? And you've heard a lot about messenger RNA because of the messenger RNA vaccines, Pfizer and Adreno. So mRNA stands for messenger RNA. So how do you get from DNA to messenger RNA to a protein? Well, this is a very simple ABC thing. The first step is transcription. What happened? Remember that double-stranded DNA? The strands pull apart from one another. The bases separate. The complementary base pairs separate, creating a gap between the 2 strands. And on the exposed sequence of bases now, other bases that might be floating around in the fluid but nearby attach themselves and form a matching strand. That's the messenger, that's a single strand of RNA. So its sequence reflects the sequence of the gene, or at least part of the gene. In mammalian cells, that messenger, the DNA is in the nucleus. So that messenger RNA has to find its way across the nuclear membrane and into the cytoplasm of the cell, where there are other structures, and they are called ribosomes. And the ribosomes are the protein factories of cells. So those ribosomes read the code of in the messenger RNA and translate that sequence into a sequence of the appropriate amino acid. So sequential 3-base sequences of bases dictate the order and the type of amino acids selected for the protein. And most importantly, that order of and that sequence of amino acids and the choices of those amino acids dictate the function of the protein. So if there are mutant forms of, if the gene is mutated, the parent gene in the nucleus, and therefore the messenger RNA transcript of that DNA copy of the gene is mutant as well, then the protein it makes will be mutant. That's exactly what happens in Huntington's disease and myotonic dystrophy, which I've already talked about. So now on to gene editing and the CRISPR-Cas technique, the technology that won a Nobel Prize for Emmanuel Carpentier and Jennifer Doudna last year. It was really based on Emmanuel's work or contribution to this, came from her work on a bacterial immune system for destroying viruses. So bacteria regularly encounter viruses, and when they do, sometimes it's not good for the bacteria. And viruses, and if they're vulnerable, maybe the majority, maybe 99.99% of those bacteria might be destroyed. But if one survives, some part of that virus's RNA or DNA is copied into the bacteria's DNA. And so that bacteria now has a memory for that previous infection that it survived. So if that bacteria runs across the same virus or a very close relative of it, whose code is very similar to that coded in now its own library of previous encounters with viruses, if there's a match, then that bacteria can destroy the invader. Now that's the principle of CRISPR. So on the left, there's a cell simplified. There's a nucleus. It shows 5 chromosomes in it, the cytoplasm out here. And let's say on one particular chromosome, there happens to be a mutant gene, an abnormally functioning gene. And the goal is to snip it out and replace it with a healthy gene. How would you go about doing that? Well, remember what I just talked about, the bacteria's tool for dealing with viral invaders. One is that you create a guide molecule, an RNA guide that might be 10 or 20 or 30 bases long, but anyway, a guide, not to the whole gene, but to part of the gene, so that guide will find that gene. It will look for that sequence once it gets into the cell. And then another protein that's very closely attached to that guide has an enzyme properties. That is to say, it can break or destroy, cleave, cut, those are all terms, DNA at that site, at the site of the guide. So you have a guide, it has an address, it knows how to get there, recognizes the address. I can do that for you. The best, easiest would be for you to register yourself. And attached to that is a DNA cutting enzyme. If you go to the web, so here we are, move it over to the second from the left here. here's the
everything is on
guide
our website.
designed to match a sequence on all of the gene, or part of the gene here, in the DNA that you want to fix.
So it's a three-part
And
series.
so with the guide guiding the enzyme, the protein, to the
So
site,
it's
that enzyme then cuts the DNA.
Yeah, they're all on Tuesday
And
night.
then if you happen to have
So
on
I've got,
board
yeah, so
a
we've got
normal
one on
copy
October 12th, one on
of a
November
normally
9th,
functioning
and
copy
then
of
the
that
last
gene, you
one
can
will
insert
be
it. So here's a
December.
way of um, identifying and finding the address of a mutant gene,
6:30.
cutting out the abnormal gene,
Yeah, yeah. So
and
it's 6:30
inserting
and then
a
the last
normally
one will be
functioning
December
copy.
14th. So it's
That's
6:30
what the
at
Nobel Prize
library.
was
It's going
all
to
about last year.
be in person, obviously we're limiting numbers and we'll have to be socially distanced.
Well, how do you go about doing this? Well, there are
But
lots
they're
of
also
ways,
live
and
streaming.
I can't address them all in this talk, but here are some thoughts about it. One way is that you develop the guide, and by the way, these guides, if you know what the target sequence happens to
Yeah,
be,
so you just
commercial
automatically get
companies
registered for
will
all
produce
three if you can't make it.
the appropriate RNA guide for you.
So that's why we just
And couple that with
So I've got
the
I've got it
enzyme
up now. So
to
you want
chew
to
up
do the
the
first thing?
mutant gene that you want to correct here. And then
Last thing.
you could package that in a virus. The commonly used one is, or one of the adenoviruses. These are kind of these are viruses that can't multiply once they get in the body, but they're simply being used as a vector, as a way of getting the CRISPR and the attached enzyme into the body. And if you inject it into the circulation, it has access to all of the cells in the body. But you may want to direct that more specifically to an organ or to a specific tissue. So you may want to inoculate the liver if it happens to be a mutant gene in the liver that you want to go after. Or if it happens to be a muscle disease, inject it into the muscle.
Is anyone going to be
Now,
joining you?
another way of doing this, and this is most easily applicable to bone marrow disorders, meaning diseases that are related to red blood cell manufacture, or white blood cells. You might remove
Yeah, no, I don't have any. I don't have to get any other. For me, it's just going to come
stem
as
cells
gas
from the bone marrow,
as
and then do the editing
what numbers these days? Okay, so you
side
should
of the
get
body.
a confirmation email.
So you fix the cells
And then you should
outside
also
of the
get
body.
my emails as
At the
Dave
same
David.
time,
Yeah,
you
hopefully that's for you.
using
We'll see you then.
chemotherapy,
You're welcome.
you might try to destroy the previous occupants that of those types of cells that you're going to replace before you then transplant the fixed or edited cells here. So there's another way of doing it. Now, I'm going to walk you through this next one. This is an uncommon disease. It's a mouthful. I don't expect anyone to remember, but follow me as I walk you through this. Here is a paper using CRISPR-Cas9, so the Nobel Award here for the technique, in vivo gene editing for a disease called transthyretin amyloidosis. Well, transthyretin is a protein. There is a gene responsible for manufacturing it, and that's a gene, and the production of the protein is mostly in the liver, not exclusively in the liver, but mostly in the liver. And the genetically transmitted form of this disease is autosomal dominantly disorder, and it's rare, but a much more common one. A mutant gene may appear in a person in the lifetime. So that mutant gene may be acquired, and that's called wild form. But the point is that this mutant gene produces a mutant protein and a mutant version of this transthyretin protein. Now, the normal transthyretin protein is made up of 4 components, tetramer, word here, and the molecule is soluble. But in the mutant form, it's insoluble, and it creates deposits of insoluble amyloid fibrils, and they build up in the peripheral nerves and cause a peripheral neuropathy, and if they build up in the heart, it causes progressive heart failure, and these deposits can occur in other tissues. By the way, that's this buildup of abnormal amyloid in the heart is a common cause of heart failure. Well, one way of dealing with this problem is, well, there are 2 options here. Fix the gene or go after the messenger RNA. Remember, messenger RNA is exactly what its name implies. It's the messenger boy between the nucleus, between the DNA, between the gene in the nucleus, and the protein manufacturing machinery in the ribosomes in the cytoplasm. So one way of treating it would be to do something to the messenger. So you can block the effect of this mutant gene by blocking the effect of the messenger RNA. And there are 2 drugs now have been out now for in trial, and they're now approved, and they work. They can certainly stop the deposition of these insoluble amyloid fibrils in nerves and the heart, and maybe even reverse it, particularly if they're treated early, if patients are treated early. But the problem is these treatments involve repeated infusion, perhaps monthly infusion, and it's expensive. So it might cost anywhere from $50,000 to $100,000 a year. So not only is it inconvenient, but it's expensive. Well, why not? The option, of course, is go after the nuclear mutant gene, the mutant gene in the DNA. And so the CRISPR guide here has been designed to target the abnormal TTR, if you like, gene, and specifically a sequence. And that's the string of 20 bases. That's what it would look like. That's the string of bases in the CRISPR guide. That's the address for the gene. It's not all the gene. It's only a part of the gene, but it's enough to target that specific gene. That CRISPR guide is coupled with a protein with enzymatic properties, so it can snip the strands of DNA, and the whole is packaged in a lipid nanoparticle delivery system designed to target the liver. Now, this is exactly the kind of transport system that was used for packaging mRNA in the Pfizer and Moderna vaccine. If you just infused or injected mRNA, it would be quickly destroyed by circulating enzymes. So it needs to be protected. So that's why it's been packaged in a lipid shell for delivery. And you can also engineer into that package a destination address, meaning most of it will go to the liver. The beauty of this technique is it's a one-time intravenous infusion. So let's just move along. Now, this slide is kind of crowded, but just to show you what these lipid shells look like. Now, this happens to be what kind of a cartoon here of what this lipid shell, this outer shell, would look like. And inside it, in this thing for the thyretin gene that we're trying to target, here's the guide, here's the enzyme. So the guide and enzyme coupled together, protected in this fatty shell. Then that's infused. Here's this intravenous bag here. And it heads right to the liver. And when it gets to the liver, here it is, it's kind of moving along in the capillaries. Then it moves across the gap and inserts itself into liver cells. Well, they're called hepatocytes, but liver cells. And once in the liver cell, it gets it the whole bit. Now, this is greatly magnified here, but here's the Cas. Here's the Cas enzyme here that's going to chew things up. There's the guide here, and it's found the address of the abnormal mutant gene, and it chews it up. It destroys it. In this case, it's a normal version isn't inserted, but it's enough to protect the patients against the abnormal protein. And you can see that in the next slide. Here's the drop-off in this production of the abnormal protein, which is causing the peripheral neuropathy and heart failure and the other symptoms related to this disease. And here's the rise in this mRNA expression here. So it's very, very successful here. The most important curve is this one. It's showing that precipitous drop in the mutant gene, and therefore the mutant protein causing the disease. So that's a beautiful example. Now, I alluded earlier several slides ago, and there's a similar system, a similar viral delivery system for DNA that was used by AstraZeneca and at the Oxford group for the AstraZeneca vaccine. So one way to insert genetic material or the CRISPR, the guide, and the enzyme, either one, either the CRISPR and the enzyme or the corrected gene, is to package it into a virus that also protects it from the moment it's injected into the subject to the moment it gets into a cell. Once into the cell, it releases whatever package you wanted to deliver. So whether that was the already edited gene into the cytoplasm of the cell, or whether it's the CRISPR and its associated guide to fix the abnormal or mutant gene. So viruses. By the way, these viruses are all, there are two important things to know about them. They are modified so that they cannot multiply. So once in there, and once they've done their job, they're gone. And I should add one thing, and here one of the things that may have caused some of the trouble with the AstraZeneca that vaccine, that this is an aside comment, but it may have been the viral vector may actually provoke an immune response, and some people have wondered whether that was responsible in some way for those thrombotic events, those rare thrombotic events that made many people reluctant to take the AstraZeneca vaccine, and some countries not to use it. Anyway, back to these viral vectors. Well, here's an example, adrenoleukodystrophy. That's another rare in this case, autosomal recessive disease, meaning that both parents have to carry the gene or complements of the gene or copies of the gene. And or maybe just the female carries the gene in this case, but it's on the X chromosome, as it is in this case, and so it is the males that are affected. In any case, adrenoleukodystrophy is a nasty disease, and it causes paralysis and affects cognitive function. It's not a good disease. But one way of fixing it is to harvest stem cells from the patient's bone marrow, and then outside the body, infect it with viruses, these tame viruses, but tame viruses that are carrying healthy copies of the mutant gene that's causing the adrenoleukodystrophy. Once inside the body, those cells find their way into the brain, where the correct version of the gene is inserted into nerve and other related cells, glial cells, to prevent further pathology and clinical progression. This works. Surprisingly, it works. But it works best if you catch patients very, very early in the disease. Now, here's another rare one, but it illustrates several points. It's another one. Here we are, gene therapy for aromatic L-amino acid decarboxylase deficiency, or for short, in this acronymic universe, AADC, by direct MRI, guided direct delivery of the normal gene to the brainstem. So this is a rare autosomal recessive developmental disorder. It's caused by a gene responsible for production of an enzyme, which is necessary to change a precursor molecule into dopamine or serotonin. And those are two of the most important neurotransmitters in the brain. The absence of those transmitters, dopamine and serotonin, causes a progressive disease, manifests very early in life, manifests by motor dysfunction, loss of tone, and oculogyrate crises. The eyes will suddenly go up together or off to one side or the other side, and sometimes they're stuck there for hours on end, and it's an uncomfortable symptom. And they often experience trouble sleeping. And it isn't long before they're unable to sit up, stand, walk, or even have trouble swallowing. So it's not a good disease. So what's the treatment here? Well, a viral vector that expresses the gene that is responsible or codes for the protein and enzyme necessary to create dopamine and active dopamine and serotonin. Now, in this study, they went after dopamine. They didn't try to correct serotonin at the same time. But so let's go back here. So what they did neurosurgically with a stereotactic equipment, so they had 3D control, they injected these viruses carrying the appropriate, normal, properly functioning gene into the brainstem. That would almost be unthinkable for me even a few years ago. But it was very successful, and there was no difficulty doing it. And they inserted it into very small structures in the brainstem, the upper brainstem. And they were able to track their success And here are pictures of, these are horizontal pictures of one subject, the brain, at two different levels here. And it's probably too faint for you to see. I could barely see it here with the screen right in front of me. But boy, they hit the target right on here for the substantia nigra. And but look at what happens 3 months after. 3 months after. I think you probably can see here using the tags to light up the dopamine here, so you could actually see it. Boy, so both areas in the substantia nigra now are loaded with dopamine. And if you look at the level up where you can see the upstream related basal ganglia, look at the spread of the dopamine up into a structure which we call the striatum. That's amazing. That is absolutely amazing. And so what was the effect? How did this all turn out? Well, they had 7 children, very young children. So they showed that direct infusion, that injection, was safe, was well tolerated, and it reached at least 70% of the intended territories in the brainstem, with no spillover effect in the adjacent areas. 2, dopamine metabolism increased in all 6 candidates in the midbrain where the injections were actually satisfactory. Those oculogyrate crises, they resolved in 6 of the 7 candidates. 6 of the 7 candidates regained head control and were sitting independently. And a year and a half later, 2 of the subjects could walk with some support. That's amazing. That is really, talk about miracles. We'll maybe come back to this in that November talk, but that's a miracle by anyone's standard. Now, we're getting to the end because I want to talk about disorders of red blood cells. Now, these are not rare disorders. At least sickle cell disease and beta thalassemia are common disorders. Sickle cell disease, especially among African Americans and Africans, and beta thalassemia in Africa as well. Hemophilia is not all that common, but it certainly, boy, has that ever changed, the management of that disease in the last 20 years. So let's have a look at this. Now, this is a very busy slide here, so bear with me. What happens with thalassemia is that the gene, the HBB gene here, don't you love these acronyms, creates a fragile red blood cells. So they have a shorter shelf life. They don't last as long. So these patients lose red blood cells, but they need transfusions all the time. And transfusions are fine in the short term, but when you have to keep doing that, what happens is that some of the iron becomes deposited in other tissues, and that causes its own medical problems, which I won't get into. But that is an issue. And limiting on how long you can continue and how frequently you give transfusions or symptomatic treatment here. And the same transfusion dependence also occurs in sickle cell disease, and it gets its name because another mutant version of the same gene causes the red blood cells to literally crinkle up or sickle up. And that can sometimes destroy or at least block blood vessels and cause strokes. It can cause, so it can block the circulation in small blood vessels and larger ones. But it's characterized by a lot of pain too, severe pain. And it can be a miserable existence. and it also shortens life, just as beta thalassemia may. So, one thing that you should realize is that the hemoglobin we are born with and we have in utero is not the hemoglobin we carry into adulthood. So there's a fetal form of hemoglobin, and there is an adult form of hemoglobin, and the transition takes place sometime after birth. So children with thalassemia and sickle cell disease are not symptomatic in utero or early in life. They become symptomatic with the transition to the adult form. So one strategy has been, well, if the fetal form of hemoglobin is so good, or at least is compatible with, much more compatible with a healthy, normal life, why not block the production of this abnormal adult hemoglobin and favor the production of fetal hemoglobin? Well, there is a transcription factor. It has a fancy name, BCL11A. It's a transcription factor. It's a protein thing. Get back to, I think, slide 8 or 9 in this series. And that transcription factor is responsible for turning off the formation of the fetal form of hemoglobin and turning on the production of the adult form of hemoglobin. So one strategy would be to block this transcription factor to enhance fetal hemoglobin production and block the abnormal mutant adult form of hemoglobin. So that's the idea. So I hope you're still with me. To restore the phenotype of this hereditary persistence of fetal hemoglobin, you could use CRISPR-Cas9 gene editing. And as a way of reducing the expression of that transcription factor in bone marrow cells and restoring fetal hemoglobin synthesis. So that's the way to go. Sickle cell disease, I don't think I need to say anything more about that, but you can use basically the same technique or a very similar one to favor fetal hemoglobin production and block, at least as much as possible, the adult one. Now here's one of these cartoon things which illustrates how this is done. This is a patient with thalassemia, beta thalassemia, but it could be a child with sickle cell anemia. So you harvest stem cells from the blood, from the peripheral blood, or sometimes the bone marrow, and then outside the body, modify those cells using CRISPR to block that transcription protein and to favor the production of fetal hemoglobin as against the adult form. And then you multiply those cells and then you reinfuse them. So again, it's a one-time fix. It's a great way to go. Now, just switch here, we're getting near the end here. Hemophilia. Well, a little bit of history here. The hemophilia B or factor clotting involves a bunch of factors. And the ones we're going to be talking about are factor 9 and 10. Well, factor 9 this could be traced back to Queen Victoria, who's the most famous carrier of hemophilia B, who had at least 9 sons, grandsons, or great-grandsons affected with hemophilia, known as Christmas disease, after a 10-year-old boy, Stephen Christmas. It causes bleeding into joints and muscles, prophylactic infusions of factor IX, it works, but these are infusions that might be weekly or every 2 weeks, has an annual cost in 2011 days of $300,000 a year, so it works. It's very costly. Well, here's a fix: a single intravenous injection of an adenovirus associated virus vector, which is engineered to express the gene for factor IX. So that's the idea. You've incorporated the gene that's responsible for producing factor IX. You've incorporated that in this adenovirus, and then you infuse it into the subject. It works. It works. So no more infusions every week or every 2 weeks, and no more hemophilia. No more hemophilia related crises. It's a great solution. And I'll just close on one item here. There are several genetically transmitted diseases that affect our retina, and some of them are modifiable. And here's the technique. It looks awfully painful, doesn't it? With this long needle being stuck into your eye, and really inserting this package, this viral vector, carrying the gene fix into the retina, and it restores more or less, maybe not normal function, but a lot better function than before. It stops the progress of the disease. So that's it. Now, nothing, just the last slide. My two favorite players in tennis, they were missing at the US Open, both with injuries. And anyway, it brings up this whole question about the ethics. Is it possible to modify any mammal, or human to prefer certain or to acquire certain traits? It might be behavioral traits, it might be cognitive traits, it might be height, might be muscle development, might be quickness, athletic skill, etc. Is it possible? Not quite yet, but for sure within this century, and probably within the first half of this century. The technology is moving along that quickly. But as I showed you, I think the second slide here, people are concerned about the ethics about that. But I can assure you that there are several countries in the world that aren't the least bit concerned about the ethics of this. Will it happen? It will happen for sure. It's already happened in experimental animals. So that's the talk. I've enjoyed putting this together. I hope it wasn't too difficult to follow, but just remember, turning the corner here to COVID-19. Wasn't it amazing that that very effective vaccines appeared within months of the outbreak, the finding out about the outbreak in China in late 2020? Sorry, 2019. So isn't that incredible that just within months? Well, let me tell you that that the technology for those very effective vaccines, the messenger RNA ones, the Oxford one, and other similar vaccines, all that stuff was off the shelf stuff. People, for example, the Pfizer vaccine, work had been going on in BioNTech, small German company, run by a husband and wife team, who might well get a Nobel Prize, if not this year, then next year, and or the pharmaceutical companies involved. But they had been working using mRNA technology, and that and in the development and the use of that lipid protective shell to protect the messenger RNA on its way to the host cells. That's the key, putting those two together. It wasn't much of a trick to put a patch of RNA in a similar shell to protect it from the body's enzymes until it got into the cells, and that patch of mRNA or RNA coded for spike proteins and the COVID-19 virus. So all of that technology was really off the shelf stuff. Probably most of the time was taken in scaling up to produce hundreds of thousands and millions and now billions of doses. That was really extraordinary. But the science behind it already existed. It already existed for at least a decade or more. But still, a remarkable tale. So thank you very much. Now, next month, we're going to talk about that very important subject of a drug to actually treat Alzheimer's disease that might be effective. We will see. So I will see you next month, and probably using the same format. Thank you very much. I hope you enjoyed it.